Hello people, welcome to the channel. My name's Vincent and I'm coming to you from the UK. In the following presentation, Professor Thomas Seyfried discusses the greatest tragedy in the history of medicine, and he proves without a shadow of a doubt that the gene theory of cancer is wrong. He then describes the future of cancer care and how, without toxicity, we can manage the disease. Many of his papers can be found on our website metcancer.com, and he challenges you to prove him wrong. Please share this video far and wide because this message needs to get out, especially to doctors and oncologists. Please give it a like and don't forget to subscribe to the channel.
Pleasure for me to be here this evening and have the opportunity to share with you the work that we've been doing on cancer and how we plan to manage this disease. If I can get my program going here, I hope it's okay. Well, this is the building, our main campus building at Boston College, where I am a professor in biology, and I teach cancer biology and general biology to the freshmen, so I have quite a bit of student experience in that regard. So what I'm going to do today is I'm going to give you an overview of the situation of where we stand with cancer and how we plan to manage this disease. So I start my lectures with a report card from data from the American Cancer Society, and they log this information for us every year. So this is the evidence. I've gone back as far as the 70s, but right now we're looking at just the last 5 years when we've supposedly made so many major breakthroughs that you hear about on the news seems like every month. So what we have is the year 2013 to 2017, new cases, deaths per year, deaths per day. The numbers are quite sobering. As you can see, the percentage increase, we have over 1,600 people a day dying from cancer. These are not good numbers. These are the data. These are the numbers that you don't hear about on TV when we hear about all the great things that are happening. And if you notice the percentages, the deaths per day or deaths per year is increasing faster than the number of new cases. And if you want to put this in perspective, the percent of population increase over that same 5-year period was about 2.9%. So how's this war going? Abysmal. We're failing, all right? Don't let, if people say we're making all this progress, there's no progress, all right? Otherwise, we wouldn't have these numbers. So now you have to ask yourself, what do we want to do? Cancer research. What cancer research, right? We got pink ribbons painting airplanes. We have people running for brain cancer, swimming for this, jumping for that. Let's raise, do you ever ask where the money goes? What are they doing with all that money? Billions of dollars in cancer research. What's going on with that? We got a lot of dead people here. The more money we raise for cancer research, the more cancer we get. There seems to be no accountability with what's going on here. It's a tragedy. Now, the reason for this, in my mind, comes from a fundamental misunderstanding of what the nature of the disease actually is. So conflicting views on the origin of cancer is, in my mind, responsible for this disaster that we call cancer. So is it a nuclear genetic disease, as the dogmatic view will tell us, or is it a mitochondrial metabolic disease, the evidence of which I will show you? Because we have mutations in the nucleus in many cancer cells, but not all cancer cells. There are some cancers that have no mutations in the nucleus. People don't talk about that. But every tumor cell has defects in its respiration, the little bean-shaped mitochondria that you see there. It's not actually a bean, it's a spaghetti network, and when you cut through the cell, you get those little parts of the spaghetti, and that's what these bean things look like. They're actually a tubular network inside the cell. So there's abnormalities in both of these organelles, and there's abnormalities in some other organelles, but the majority of people focus on the nucleus. So, and this is the dogmatic view that we have, the orthodox view we have today. Cancer is a genetic disease in this paper by Hanahan and Weinberg, which is one of the more highly cited papers in the entire cancer field. summarizes all of these particular hallmarks, and one of the statements in the paper, cancer carries the oncogenic tumor suppressor mutations that define cancer as a genetic disease. And as I've said in many of my YouTube videos and lectures, a dogmatic view is a dogma is an irrefutable truth. A dogma is when you have a religious belief, it's a dogmatic belief. And don't think science is very similar to this. You spend your whole life studying a particular thing, it becomes a dogmatic view, it's very hard to change. It's irrefutable truth. How do we know this? We know this because I teach these classes, and every book in biology, biochemistry, and cell biology, in the cancer section, cancer is a genetic disease. You go to the NIH website, cancer is a genetic disease. There's no discussion. Well, maybe it might be sometimes and not all the time. No, it's a genetic disease. So what this dogmatic view has done, it has indoctrinated several generations of physicians and scientists into this view that this is the way it is. So to educate the next generation about this, we have these little pictures, and this is what a biology textbook looks like. Somatic mutation theory is the foundation upon which the gene theory is based. Somatic mutation theory. Mutations in genes that inhibit or stimulate cell division. And you can see the cells there, the division. Cancer is cell division out of control. What is cancer? These are cells that divide out of control. What's making them? Well, we have defects in oncogenes, which makes them grow faster, and they also have defects in tumor suppressor genes. They mean no breaks, they can't stop it if they wanted to. So we have genes that make them grow fast, and we have other genes that can't stop them from growing, and this is suppressor genes and oncogenes, and this is all we hear about. And you look at these little pictures here and it tells you. Then we have another picture to kind of familiarize people with this dogmatic view. We say, well, we have a normal cell here, the normal cell, and it has to go through a series of mutational changes, and then it becomes this malignant mesenchymal dysmorphic cell. I think everybody can see it right here. So that's the so-called tumor cell. And the question is, well, how do you get these? Well, we collect the mutation. How many mutations? 1, 2, 3, 4? I don't know. Sometimes 1, sometimes 2. What about the tumor cells that have no mutations and yet they're cancer cells? Well, we don't talk about those. And then there's other people who say, well, we can get several million mutations. And if you look at the data from the genomic projects, one mutation piled on others. We've got drivers, passengers, every kind of a name you can think of for a kind of tumor mutation. And this is the way it is. And then where we are today, we have what we call precision medicine. You've heard it on the TVs, personalized cancer treatment centers of America, cancer treatment centers of America. We're going to use personalized medicine. Dana-Farber in my neck of the woods. Yeah, we're going to use personalized therapy, precision medicine, and all this kind of stuff. So what you have is this is where it is. So this woman now is looking into the screen, and she's looking at different kinds of overexpressed genes, trying to make a decision. Can we give a prognosis, or can we recommend the treatment based on this? Now you have to realize, where are they getting this information from? They take a punch biopsy of some part of your body, where this is breast, but you could be taking it from the colon, from the liver, or whatever it is. And then you take this and you spread it out and you get these spots. Now you have to realize, I have here observer effect, when you take a lump and you stick it with a needle so that you can identify what kind of mutations in there, you already changed the microenvironment of that lump. All right, it's called inflammatory oncotaxis. So the information she may be looking at could be completely different now in that region based on the fact that she stabbed you, right? Take this thing out, and we're looking at this. Now this would be okay if the information on that screen had any redeeming value to your health. The answer is it doesn't. And it costs you $7,000 to have that woman stare into that screen to tell you. $7,200. And I read in the Wall Street Journal coming down the plane that Roche Holdings, one of these big pharmaceutical, just bought the company in Boston for $2.4 million that supports this kind of stuff. And I'm saying, wow, they really don't understand the nature of cancer, do they? So, but anyway, they think they're doing this, and I'll show you evidence that argues. So, when you argue against some dogmatic view, I don't care what it is, a religion, a political affiliation, a scientific view that's different from the prevailing view, it's always, I don't want to look at the data, I don't want to talk about it, and I don't want to hear about it. Basically, this is what we're up against. when you introduce something new. So let's look at just a brief snapshot of some of the evidence. This is a key experiment published in the top journal Science, 1969. I had a chance to discuss these data with Dr. McKinnell. There's a frog that has kidney cancer. It's very lethal, kills these frogs. It's division out of control. It's a well-described cancer. Now, mammalians or amphibians, you can do some really nice, nifty biology. So what Dr. McKinnell did was he took the tumor out of the frog and then isolated a tumor cell from the frog's tumor, and then took a fertilized egg and removed the nucleus of the fertilized egg and put the nucleus with the tumor suppressor and oncogenes into the new cytoplasm that contained normal mitochondria, and they cloned the frog. The frog developed, this tadpole developed from the nucleus of that kidney tumor, right? The tadpole is fine. They looked at that tadpole in every way imaginable a tadpole could be looked at, and they didn't find any dysregulated cell growth. That's the signature feature of what the issue was, though, there was something in that tumor nucleus that prevented this tadpole from becoming a mature frog. So there was some defect in the nucleus that blocked development, but it didn't cause cancer. Interesting. So many years later, Rudy Janiss's group at MIT, Rudy is probably the premier developmental biologist on the planet. He looked at all those other data and he said, listen, I don't believe any of that unless I do it. All right. So he did it, and he did it with mice. And he took nucleus out of a melanoma cell, and he characterized all of the mutations in the nucleus of the melanoma and then put it into an embryonic stem cell and cloned the mouse from the nucleus of the melanoma cell, right? So the presence of major genetic abnormalities in the embryonic mice cloned from the tumor nucleus provide unequivocal genomic evidence that the mice were cloned from the nucleus. But these mice did not have cancer, and they died, they aborted. Just like the frog couldn't develop to a mature frog, these mice couldn't develop beyond a certain, and it depended on the kind of mutations that were in that nucleus to determine whether or not the mouse survived. So clearly, these mutations were disrupting development, but they weren't causing cancer. Now here's another nice paper by Benny Kiripatou and his group at Baylor College of Medicine, and he left the nucleus in place, but he changed the mitochondria. So what he did is he took a highly aggressive metastatic breast cancer and removed the mitochondria that were in the cytoplasm. These are hard experiments. These are much more difficult than the nuclear transfer experiments. But he put the mitochondria from a normal cell surrounding the nucleus now of the tumor cell, and these tumor cells regained their growth control. They didn't form cancer, they didn't spread, they didn't do anything. So it was clear that the mitochondria were calling the shots, not the nucleus, okay? So I wrote a paper, in fact it was chapter 13 in my book, and I upgraded it to this paper that was published to Open Access Frontiers in Cell and Developmental Biology. And in this book, in this paper, what I did now, I don't have time. I could spend the whole night telling you about every single experiment that challenges the gene theory. And you'd be saying to me, enough, okay? Read the paper, and then you will have all the evidence. And don't listen to what anybody else tells you, all right? You can think. People can think, I don't want to believe, I can't be right. Read the damn paper, and you make your own decision. You know, I can't believe so many people they take secondhand information. So now here's the summary of that. And green cell is normal. Normal cells beget normal cells. The red cell is a tumor cell. Tumor cells get other tumor cells, right? So what's responsible for the neoplastic tumor phenotype? Is it the mutations in the nucleus? Or is it the defects in the cytoplasm and the mitochondria? Because they're both defective. The nuclear transfer experiments were able to tell us the answer to that, because when we took the red nucleus and put it into the green cytoplasm, we got normal cells, normal tissues, and sometimes a mouse, or almost a complete, without cancer. Interesting. But when we take, Israel and Schaefer did the other one. They took the nucleus of a normal cell and put it into the cytoplasm of a tumor cell, and they either got dead cells or tumor cells. They did not get normal cells. Clearly, it's the mitochondria. These are the strongest evidence to date to disprove the gene theory of cancer. The entire cancer field is based on the gene theory, precision medicine, right? Think about it for a minute. Opdivo, Keytruda, CAR-T, all that's based on the gene theory. If the gene theory is incorrect, What do you think the probability of those therapies having any real redeeming value for the majority of people that have cancer?
0.
Right? You look at the dead, look at the numbers of dead people. That tells you when something works, we drop. Just like the AIDS, when they came out with the protease cocktail, the death rate went down. That worked, right? What we have here is something that doesn't work. So if somatic mutations are not, how do you get cancer? Where's this coming from? Warburg had this identified a long time ago. Otto Warburg, German scientist, the leader of one of the thinkers and doers of the 20th century. Cancer arises from damage to respiration in the cell. Energy through fermentation compensates for insufficient respiration. So I'm going to talk more about this. And then you hear about this Warburg effect. Well, we've just recently identified the missing link in Warburg's theory, and that is mitochondrial substrate level phosphorylation. That will clarify this for people that actually can read the literature and understand what's going on. So let me ask you this. If enhanced fermentation is the signature metabolic malady of all cancer cells, and every cancer cell in the tumor has different gene mutations, and every cell in the tumor is different from any other cell in the tumor with respect to its genetic mutations, does it make more sense to focus on the unique differences of every single cell in the tumor or the common problem that's existing in every cell in that tumor? You'd think we would go for the commonality, right? Wrong. Let's focus on the unique mutations in every one of those cells and see if we can manage the disease. How's that working? Not good. So now we look at the energy metabolism in our cells. Okay, this is taken again from college textbooks. I don't want to go through if you want me to go, I can go into great depth, but for the sake of time, I don't want to do that. So all we have to do is look at the red starbursts, the yellow bursts. Okay, so we have glycolysis in the cytoplasm. It gives us a little bit of energy through an ancient pathway called substrate level phosphorylation. The Krebs cycle, an ancient pathway, gives us a little bit of energy in the mitochondria TCA Krebs cycle. But the majority of the energy that we get in our body comes from oxidative phosphorylation. How do we know that? We're breathing. Anybody here not breathing? Any zombies out here in the audience? No, we breathe air. And we exhale CO2. And that's because we're respiring. And we're getting a buttload of energy from that. And that's what's keeping us, brains, our intestines, everything that we do, muscles, they're all using oxidative phosphorylation. Now, when we look at cancer cells, if you can just look at the shift. Okay, very little energy is coming. These cells are not breathing. They're fermenting. They're using energy from these ancient pathways to generate the energy for growth. All right? So this is the shift. The tumor cells have shifted away from oxidative phosphorylation to fermentation. They shift because they're damaged. The respiration is damaged. So that's the key shift in what all cancer cells are fermenters. Regardless of their genetic mutations, they're all fermenters. So people ask me, how do we get cancer? We can get cancer from any number of different provocative agents, okay? So we know, why do they call a certain chemical a carcinogen? Because it causes cancer. How do you know that? Well, if I put the carcinogen on the ass of a rat, a tumor will form. Okay, that tells me that thing is going to cause cancer. All right? So we're exposed to carcinogens. Radiation. People fear radiation because it could give them cancer. Unless you have cancer and they irradiate you. Right? Then you have hypoxia. So a small interruption of oxygen in a particular tissue, a milk gland of the breast or some other part of the body, boom, you can damage the respiration. The cancer cell then must ferment, and I'm going to go through this. Inflammation, systemic inflammation, rare inherited mutations. PL has to be genetic. We got BRCA1, we have P53, Li-Fraumeni, all this other stuff. Yes, they will cause cancer only if they damage the mitochondria. Because people walk around with BRCA1, they never can't get cancer. Why? Because the mutation never damaged their respiration. Therefore, these are secondary causes. The primary cause is damaged, and that's what happens to viruses, RAS oncogene, age. The older you get, the more likely your respiration will be damaged, the more likely you're going to get cancer, just because you live older. You're older, right? So we call this the oncogenic paradox. How is it? And this is what perplexed the field of cancer for decades. And everybody who read Emperor of All Maladies by Sid Mukherjee, Pulitzer Prize winning book on New York Times bestseller for years, He wrestles with this. If you read the book, write down page 208 and 303. We don't understand how it's possible that we can get cancer from all these different things through a common pathophysiological mechanism. And that common pathophysiological mechanism is damage to the respiration that causes reactive oxygen species, and they're very radical. They damage proteins, lipids, and DNA. And you get the mutations coming in the nucleus as a secondary effect of the ROS coming out of the damaged mitochondria. And it's a cyclic effect because the more ROS, the more mutations, the more damage. And look, the green line is us breathing. Our cells are breathing. Our cells are breathing. But as they're damaged from one of these provocative agents, they start to ferment. And you can only get a cancer cell from a cell that has the capacity to ferment. If the cell can't ferment, you don't get the cancer. This is why we have very little cancer in heart and in neurons of the brain. These cells can't ferment. They die. So you have to have cells that can ferment, and those are the cells that will give you the cancer. Epithelial cells, a lot of these different kinds of cells. So we already know about all this stuff. Now we just have to simply link all of this stuff together with the hallmarks of cancer from Hanahan and Weinberg. We have tens of millions of dollars that have been spent documenting the hallmarks of cancer. So that's not wrong information. It's just that it's misinterpreted. So we have to know how to interpret all this massive stuff that people have been, all this cancer research that people have been doing, self-sufficiency of growth signals, insensitivity to anti-growth, limitless replicative potential. When that mitochondria goes out, the cells enter their default state. This is the state that all cells on the planet were in before oxygen came into the environment. Don't forget, the Earth had living organisms on this planet when there was no oxygen in the atmosphere. Those cells were all fermenters. They would grow dysregulated, unbridled proliferation until the fermentable fuel in the microenvironment was depleted, and then they would croak. And we have the skeletal remains of these things to prove that. So that's a clue. If the cell ferments and you take away the fermentable fuel, the cell dies. Not that complicated. I'll show you the evidence for this. Now, another key kicker in this whole thing is metastasis. There's cancer cell spread, all right? So when you take a punch biopsy, you put that individual at risk for spreading the person's own cancer. If the tumor cells are at a particular stage and you stab them, you can spread. Not always, of course. Sometimes it's a benign thing. You can stick it all day long and won't go anywhere. But there's other people that can risk this. So how do we get metastatic? This is the key. And once it spreads, this is when you really have to deal with the spreading, and that they throw all this poison and stuff at you. So here's the epithelial cells. It could be anywhere in the lung, the colon, the breast, whatever. All of a sudden, one of those provocative agents from what I just went through causes these cells to proliferate abnormally out of control. They start to grow and they become a little bit more out of control. But they're not going to spread. They're going to stay right where they are. And the red cells are our immune system, all right? Our immune system is designed to heal wounds, all right? So we have a group of cells that are misbehaving. And they're sending out certain signals that attract the red cells that are supposed to come in to heal the wound. So they put out growth factors to facilitate wound healing. Bad. So they're not thinking. They're mindlessly going through their evolutionary divine programs in a wrong context. And what happens is it causes those blue cells to become even dysmorphic, but they can't spread. But they're starting to, now these red cells if they don't heal the wound right away, they actually fuse with each other and with some of the other cells to facilitate wound healing, right? Okay, what happens is you dilute the normal mitochondria in the cytoplasm of these red cells with the abnormal mitochondria and the blue cells, and the next thing you have is this hybrid kind of a cell that has all the capabilities of entering and exit the circulation and spreading through your body, but now it's dysregulated. The most tough cell in our body is this macrophage of these immune cells that are now ripping through. And they try to use anti-angiogenic therapy. These are the things to try to block the blood vessel. It doesn't work. It doesn't work because the cells evolved to live in hypoxia. Not going to work. That's proven to be a failure. So yeah, if you don't understand the biology of the disease that you're working on, what do you think the probability is of making any major advances? Right? You don't understand. Most of the people that do cancer work in this country, I don't give a damn what they're UCSF, Dana-Farber, MD Anderson, they're all locked into this gene theory That's absurd, right? This is the biology of the disease. You got to understand your adversary. You got to know what they eat. You got to know what they do. And once you understand that, you can kill them. And I'll show you the evidence for that. So if most cells use fermentation, what do we do? Well, one strategy is target their fermentation, all right? Not terribly difficult to understand, but apparently it is. So calorie restricted ketogenic diets, these are they lower blood sugar and they raise ketone bodies from fat which can't be fermented. So what happens is you take away the fuel of the cell that needs that glucose, and you raise an alternative fuel, which is ketone bodies, which they can't use because their mitochondria are damaged. You got to have a good mitochondria to get energy from a ketone. And these cells can't do that. So you can have, both calorie restriction. Now, I want to say calorie, I don't like the term because in humans it means water-only fasting. And whoa, I don't want to hear about that. So you have to come and we have to make a new kind of approach, which we have these restricted diets, and we want to make it fast. So I'm going to show you how to work it with drugs and things like this that have to work together, all synergistically, so we can achieve the mission. And the mission is, how do we eliminate cancer in a person's body without toxicity? That's the key. The key is we want people with cancer to be treated and to emerge from the treatment healthier than when they started. No hair loss, no vomiting, no diarrhea, none of this crap. And I'm going to show you how we can do this. So here's what ketogenic diets are also very confused. They're very low carb diets. They're higher fat, and the types of fats and the types of carbs all play a role. I don't have time for that. It's a low to moderate protein. And we restrict it. We don't go out and say, hey, just eat as much fat as you can. No, because you can get insulin insensitivity. So people have to know, ketogenic diet is medicine. All right, people have to know that. It's a medicine. It should be just as respected as much as any other medicine. So if you don't know how to use it, you're going to hurt yourself. You could. Or not work. I don't want to say hurt yourself, it might not work as well as it should. So it's a very simple thing. How do we manage the growth? We reduce the blood sugar, and we raise the ketones. And if we can get the blood sugar down and the ketones high, we know we're taking away one of the key fermentable fuels that this disease needs to grow. And working with many patients, I'm not a physician, but I work with many people who have cancer, and I work with a lot of scientists, and they feed me back, and they say, oh, my sugar is all off the planet. You know, I don't know, I got into a fight with my neighbor, my blood sugar went up, and I hear all this kind of stuff. And so my students and I sat down and we said, this isn't working. We can't have people measure independently their glucose and ketones because it seems to be too variable, capricious, and we have to have something that's a little bit more stable. So we made the glucose ketone index to give people a singular number that can give them a guide as to whether or not they're in therapeutic ketosis, because if they're in therapeutic ketosis, we know for a fact that the tumor cells are not going to grow as fast as they could if given opportunities to ferment. So we're just simply taking away one of their fermentable fuels. And we have a little calculator that we published in this paper. Therapeutic efficacy is considered best with index values of 1.0 or below. So most of the cancer patients, if they can get into that zone, and there's meters that you can get to do this. We do it with enzymatic assays, but we've done it with the meters as well. So it's a guide for the patient to say, now it turns out that so many non-cancer people with no cancer, are using the index just to see how tough and buff and all this kind of stuff they can be, which is good. I'm not arguing against that, but it was made really to help cancer patients. And one of the first uses of the ketogenic diet was done by Linda Nebling and her colleagues at Case Western Reserve in Cleveland, where they took two little hopeless cases of children that were brutalized by the system. It's unbelievable. It breaks your heart to see what was done to those kids. But she said, well, the physician said, well, they got nothing to lose. The kids are terminal, hopeless. And she rescued both kids. They both increased tremendously their quality of life, and they outlived all of the predictions, well beyond. And you know, I said to myself, man, this is unbelievable. How come nobody's doing this? They're still not doing it as far as I know. Why it works so well in kids? It really did. 'Cause I worked with epilepsy children for years. I worked on, like Adria said, we learned a lot about ketogenic diets because I worked with epilepsy for a long time. We put ketogenic diets on little kids all the time, right? But when you go to put it on cancer, oh, you can't do that. Why? You know, it works good on kids if you can do it, if you can find somebody who's not fearing going to lose their license. So here is so we said, oh, let's double check what Linda did. and see, because Linda Nebling was saying, if you got to load the blood sugar and get the ketones up, you should be able to reduce the tumor. So we developed our own glioblastoma kind of tumors in the mice. This is one of them, CT2A. And this is standard diet, which is the mouse chow unrestricted. The mouse sits in his cage, he chows down on this high carbohydrate diet. And then we gave the same diet restricted by 40% to another mouse. This is one of a group. We did many of these guys. And look at that. This is a tumor in his brain. It grows like crazy. In 14 days, this guy is history, right? They die. This guy is looking really good from the same diet, just restricted. And I said, this is before I knew what Warburg was all about, right? I didn't know. So I said, what the hell is going on here? This is unbelievable. 65 to an 85% reduction in the size of this tumor just by taking away some of the food that the mouse eats. This turned out to be the same as a water-only therapeutic fast for humans, by the way. It's not that let me go home and eat 40% less. No. You got to do it because the basal metabolic rate is different in mouse and man. So we documented each of these squares is a mouse under a different dietary condition. So the squares. So this glucose, we did linear regression. It's a statistical analysis using glucose as the independent variable and either ketones or tumor weight as the dependent variables. And you can see the squares. As blood sugar goes down, the levels of ketones in the blood go up. This is an evolutionarily conserved adaptation to the restriction of food. Happens in humans, even much better in humans than it does in the mice. As blood sugar goes down, the size of the tumor goes down. The higher the sugar, the faster the tumor grows. The lower the sugar, the slower the tumor grows. Right? Does everybody understand that? If you want your tumor to grow fast, take in glucose. If you want your tumor to grow slow, restrict glucose. Why when we go to the oncology clinics, we see the kids eating big cakes, guys drinking Infamel? Right? What the hell's going on here? These data are published, and it's not just the mouse. We have brain cancer, breast cancer, colon cancer. All these cancers have been shown to do the same thing. Why the hell are we giving the cancer patients so much sugar? It doesn't make any sense. There's some sort of disconnect. They don't read the literature, or they don't understand it. I don't know how I can make it any more clear. What do you want me to do? Am I speaking Greek? And then people say, how does it work? Oh, we published, we can't believe it unless you have a mechanism. Bullshit. We described the mechanisms over and over again. So did so many other people describe them. Oh, well, I don't know about that. What don't you know about it? Then somebody comes to me. They got a dog, right? A dog has a big tumor on his face. Look at it. It's a mast cell tumor. Here's the nostrils. It's right under his nostril. And this, of course, there was a YouTube video. I know they took it from one of my lectures, and it ran 5.3 million views on this thing. Even I'm shocked about it. So she says, oh, my dog has a big tumor. The vet wanted to take the tumor off with radiation and chemo. Sound familiar, right? Dog will live 7 months, have diarrhea and all kinds of things. Won't be very healthy. She said, I don't want to do that. She loves this dog. It's like her son. So she says, what shall I do? And she went on the YouTube, just read our papers. Like, she's not a scientist. She's just a person who owns a dog that she loves this dog. She gave him raw meat, restricted the calories by 40%. And in a couple of months, this angry thing turns into a spot, and now it's gone, and the dog is fine. This was, 2015? She just said the other day, I said, how's Minka doing, the name of the dog? Oh, he's doing fine. No surgery, no radiation. So, a lot cheaper. Yeah, she saved $10,000. Think about that, right? So now I want to talk about humans, okay? We're going to do GBM, glioblastoma multiforme. This is a real nasty, because even our Senator John McCain is now suffering with this. It has a lot of different kinds of cells. It's very aggressive. Ted Kennedy passed away from this. Most people pass away from this. I'll show you the evidence for that. It's a real tough nut. It can be tough if you don't know what you're doing. If you know what you're doing, we think we can have major, and I'm going to show you evidence to support that. Now here's a cross-section through a brain of some poor soul who died from this tumor, and you can see all this discoloration, nastiness, cystic. So, and the tumors, if you notice, this is the midline of the brain, and you can see that it should be nice and straight, and you can see it bowed out right over here. It's called midline shift. So what happens is these poor people, the tumor grows inside their brain, causes intracranial pressure, and they eventually go unconscious and they die, okay? So, And then you will say, well, we cut it out, we irradiated it, we did it. The tumor cells, they move through the brain. So even though it looks normal out here, it's laced with all kinds of tumor cells that you can't see. This is from the surgical, the radiation. Now, see these dark cells, these spaces, these are the blood vessels, and these tumor cells are using those blood vessels, those railroad systems, to spread through your brain. So this is why surgery and radiation don't work. And you can see the tumor cells around. I'll show you. So you got to realize, again, you got to understand the biology of what you're dealing with here. So now people know what I just said. They use anti-angiogenic therapies, which doesn't work, right? So here's an electron micrograph of a mitochondria, a normal mitochondria. And you can't see, you got to use the electron microscope. You can't use regular light microscopy. So these stripes that you see, these nice stripes, they contain the proteins and the lipids that allow us to breathe air to get energy from oxidative phosphorylation, called cristae. And here's the mitochondria from a glioblastoma multiforme by my colleague, Arismendi Murillo from Venezuela. He's one of the leaders in EM of brain cancer. And you can see this big wide open space that they don't have cristae. So this organelle is not going to be able to generate energy through oxidative phosphorylation. Structure dictates function. So you'll hear a lot of scientists say there's nothing wrong with the mitochondria in cancer. Are you blind? Right? Oh no, I don't think that's right. Well, what the hell's wrong with it? Look, I mean, everybody can see that, and we have dozens and dozens of articles showing this. Oh no, they can breathe. Yeah, I have to deal with this, right? So anyway, we know we have never found a tumor with the correct number, structure, and function of mitochondria. That indicates something. If they don't have the mitochondria structure and function and numbers exactly right, where are they going to get their energy from? Well, they're going to have to ferment, right? Do we know glioblastoma ferment like crazy? Most cancers are fermenting like crazy. Why are they fermenting like crazy? Because their mitochondria are defective in so many different ways. So what do they ferment? How do they stay alive, these tumor cells? Well, they have only two major fuels that will keep the beast alive. And one of them is glucose, like I just showed you, and the other is this amino acid called glutamine, which is the most abundant amino acid in the body. Cancer cells can't live without glutamine. You can't grow cells in culture without glutamine. So glutamine comes in, glucose comes in, these two fuels work off each other, and they set up a protective shield. So when you irradiate them, the shield prevents the radiation from killing them because they're using so much glucose and glutamine. So this is why you say, oh, radioresistant, we have to figure out radioresistance. It's a radioresistance because they're taking in glucose and glutamine. And they're getting all their energy, the majority of it, from substrate-level phosphorylation. And the carbons from glutamine and glucose can do everything that cell needs to grow out of control. All right? Oh, does that tell us something? Wow, glucose and glutamine are driving the beast. Okay. What happens when you take glucose and glutamine away? Well, I'll show you. But before I do that, let's look at what we do to these poor souls that go in for brain cancer, right? We published this in Lancet Oncology. Big journal, actually, in the field. So I was so pissed off, I said, listen, we got to tell people what's going on in this brain cancer field. I published it, and I thought, they were going to shoot me. Dead silence. Nobody talks about this. So let's look at what happens. Person, he might have like a seizure, or hey, what's wrong with your arm? I don't know, man. It's just, I feel a little funny. Go in, what's wrong? I don't know. They misdiagnosed. Well, then something gets happy, starts dragging. Whoa, what do we see? Maybe there's something in your brain. Ooh, we see a big tumor in your brain. Oh my God, it looks like glioblastoma. Let's take a punch biopsy. Even if not, we can look and say that's a GBM, because I have a lot of neurosurgeon friends, they tell me. We got to get that. When you get diagnosed with a glioblastoma multiforme, you are in shock. I don't care who you are. As soon as you open, what the hell is this? You're kidding me. Like the angel of death, the black spot. So, well, what do we got to take it out as quickly as possible. Oh, okay. Everybody is in shock. I don't care who. Oh, are you sure we have to? No, we got to get it out immediately. So what did I just tell you? When we surgically resect, we create a wound. All right? So that wound is already now going to contribute to some of the spread. But now sometimes, I'm not saying you don't take it out, because I have a lot of, we talk about this. If it's going to herniate your brain and kill you, you got to get it out, no question. But if you have some watchful waiting time, then you take that watchful waiting time and you apply metabolic therapy. But nobody does that. So what we do, as soon as that poor soul recovers from the debulking surgery to get rid of the tumor, which is only what the part that the neurosurgeon can see, we then bring the radiation on this poor guy, right? So we irradiate the guy, and we break apart the tightly regulated glucose-glutamine cycle in the brain. freeing up a large amount of glutamine. What did I say about cancer cells and glutamine? They use glutamine, right? So the radiation is going to free up because it creates necrotic areas in the brain. And the necrotic areas are going to free up the glutamine that I said is going to drive the beast, all right? So we're freeing up the glutamine by the very procedure that we're using to treat the patient. Now, because you irradiate somebody's head, the brain swells from the edema and the swelling from the heat from the radiation and all this other stuff. How do we reduce the swelling? We give them high-dose steroids. So we give dexamethasone, which causes hyperglycemia, right? Increases glucose. Increases glutamine, increases glucose. And to make matters worse, now that we know many of these tumor cells are infected with human cytomegalovirus, which is kind of interesting. And the viruses get into the tumor cells and they act like a supercharger so they can use glutamine and glucose even faster than what they would normally do. It's like, what is going on here? So what this therapy does, it guarantees the recurrence and death of the recurrence of the tumor and the death of the patient for the majority of people. Now, am I blowing this out my butt, or is there evidence to support what I just said? Why don't we just look at the statistics on who survives this GBM? Okay, the red line are those people, the 278 in this study. Look at what's that number there? 0. 0. Nobody survived. It's not surprising. It's not surprising. What you did to that person put them in the 0 category. Now, the biggest advance that we had in brain cancer in the last 50 years was temozolomide, a toxic alkylating agent. And they said, look at this. We can increase people's progression-free survival and increase by 6 out of 200, slightly increase overall survival by giving them a toxic alkylating agent called temozolomide. I said, and then the guys from UCSF over here published a paper in 2014 saying temozolomide increases driver mutations. What the hell does that mean? A driver mutation is supposed to make the tumor grow faster, but if I give them the same drug that increases driver mutations, I actually slow it down. Something's amiss. So what I said was, what does temozolomide do? Well, if you look at people who take temozolomide, nausea, vomiting, diarrhea, and fatigue are some of the consequences from temozolomide. All indirect forms of calorie restriction. So what is the probability that this little blip is due to calorie restriction and not the toxic drug that you're giving to the patient? Either way, it's not good. And I can tell you data from Turkey, Egypt, Germany, England, Japan, China, we get the same pattern. All right? Now let's put a face on it. This is, you remember this girl here, Brittany Maynard? She was from Northern California here. Here she is right after she got married. She was diagnosed with a low-grade, stage 1 or 2 tumor back in 2014. And within a month after they took the tumor, oh, you got a low-grade, she has a glioblastoma. Inflammatory oncotaxis. What did I tell you about that? You go in, doesn't happen all the time, but it can happen. And then, here, look at her now. She doesn't look anything here like she does here. This is moon face from high-dose steroids. Most people who have these glioblastomas, they get pounded with all the steroid, and they get moon face from all the steroids. And that steroid is making that tumor grow faster. Now, she says, I'm out of here. She goes to the northern state, Oregon, where they have a death with dignity, and she and her family decide to terminate her life November 1st, 2014. And that was January from when she was diagnosed. Less than a year, she's dead. Now, what kind of a statement does that make on a profession where you're supposed to be helping people and they're bailing out? Not good. So I'm looking. Now we have another case. This guy's Pablo Kelly from England. Pablo emailed me in the same time frame as Brittany, same year, August 2014. He was diagnosed with a glioblastoma. And he said, I don't want any of that stuff. I don't want surgery, I don't want radiation, and I don't want chemicals. So I said, okay, I gave him the stuff, and he had a YouTube showing everybody all the keto stuff that he was eating, and this thing and that thing, and the doctor said, you're going to be dead in 3 to 6 months, Pablo. You're crazy, Pablo. You know, what are you doing? This diet doesn't work. This metabolic stuff is bullshit. Pablo said, I'm going to do it because I've seen the data. So, all of a sudden, so I never heard, I didn't know. So a year goes by, a year and a half go by, and I said, oh, geez, I never. And then all of a sudden he emails me. And I said, hey, Pablo, I didn't say, hey, Pablo, I thought you would be dead. Nice to hear from you. How you doing? He said, I'm doing fine. So I said, wow, that's remarkable. He says, I think I'm going to have my tumor taken out now because they told me it was inoperable originally, and now because this diet and all the stuff I'm doing, they seem to think they can get it out, all of it. So I said, well, give it a shot. Now it's shrunken down, So he took it out just this last year. He's going to be out this summer 4 years. He's doing fine. He got married. His wife is going to have a baby any day now. And he has a very good quality of life. I'm not going to say it's perfect because he does have some seizures. But he's alive and the tumor is managed. And he has hopeful. And he's looking forward to the future, which is very interesting. So what we did is, we developed some of the best animal models of glioblastoma that there are. And we began to test various ways by which we can start managing this cancer in a more biologically friendly way, with Pablo and some other patients giving us the motivation to say, hey, we can beat this if we know what we're doing. So if we have a good tool, which is this model system where they develop spontaneous glioblastomas in the brain of this strain of mouse, we engineered the tumor cells so we can follow them using light, bioluminescence imaging, and when we treat them, we can be able to determine whether or not our therapies are going to be successful. And if they're going to be successful, we can then tell our clinician friends who see patients and tell them, this is what we think you should do, and if they do it to the patient and the patient lives a lot longer than they're supposed to, hey, that's what our job is. So what we did is we began to look at this glioblastoma in the mouse and ask, how do we stop it? So here's the tumor in the mouse's brain, and you can see it invading right through the brain. Here's tumor cells well in advance, eating a high carbohydrate diet. So as I said, you want to get your tumor to grow fast, eat a lot of sugar. So now we said, let's take away all the sugar and see if we can kill the tumor. And here's the tumor under calorie restriction, restricted ketogenic diets and everything. And you can see that invasion is markedly reduced, but the tumor is still there. So I said to my students, I said, what the hell, man? We threw the kitchen sink at that thing. We tried to take all the glucose away, and the damn thing is still growing. So I said, why is it growing? And they said, what's the other fuel? It's glutamine. So I said, wow, why don't we try to target glutamine? So we said, well, this drug has been known for decades, this drug DON. It blocks the first step in getting glutamine into the cell. So the glutamine can't get into the cell because we stop the enzyme that brings glutamine in and metabolizes it to glutamate. So we use this drug, 6-diazoxyl-L-noroleucine. We call it DON because it's hard to pronounce this. This is the structure. And we know what it can do. It can stop glutamine. So we decided to set up an experiment, which it's not published yet, but we have it almost ready. So here's what we do. And this is what I set up these scales for the mice, and we set up these scales for humans as well, because we got to know what's going on. Except we're putting the tumors into the mouse's brain. The human already has the tumor in their brain. That's the difference. So this mouse then 3 days later, it's a raging GBM, and then we put him on a fast just for 18 hours, and then we give him back the high-carb diet or the ketogenic diet restricted. So, and now they're going back to the diets, and then we pulse, bing, with glutamine, DON, bing, bing, every couple of days, bing, bing, a pulse. They have the diet. And now, of course, we stop the experiments at 15 days because the guys eating the high-carb diet are croaking. They're getting sick. We have to stop the experiment. And then we compare the two groups, the guys that get the diet and the DON versus the guys that got the high-carb diet and no DON and no diet. And then we can look at the brain. Now, this blue stuff is the light of the tumor. So you see a lot of bright light here. And that tells you that these tumor cells are actively growing, okay? So this is a high carbohydrate diet, unrestricted. Look at the light, this bright red and yellow. This means you get a lot of living tumor cells in there. This is the diet by itself. And you say, well, the diet didn't do much. I'm going to show you, it stops invasion. But you're right, the diet alone, it works. This diet works so much better in humans than mice. People say you cure these mice. You don't cure these mice. If they have the same thing that the human has, it's just as hard. So now we do the diet with the DON and there's no light. And we did this over and over again. We can't find living tumor cells, any number of living tumor cells. Wow. So I said, we got to look at the tissue to make sure we're not making any kind of a mistake. So here's what the histology, and don't forget, this is the way you diagnose any kind of a cancer. You got to look under the microscope and look at the tumor cells. And you see mitotic figures, and you look at all this stuff. Oh, you have cancer. All right, so we know these guys. So here's this line. This is the tumor, and this is the normal brain. And these tumor cells are ripping, invading right into that white area. And the tumor cells are all densely packed, growing like crazy on the high glucose diet. Look at the ketogenic diet. The line is so much more sharp. The tumor cells are not invading as fast, and there's more spacing. So the diet by itself is definitely doing something, but it's not killing all the tumor cells. When you put the diet with the drug, the glucose inhibitor with the diet, the glutamine inhibitor with the drug, we slaughter the tumor cells, right? This proves why we have no light. These are all dead cells. These are all dead, all right? The mice live a lot longer. They look healthier. So we said, we got to put this together in something that humans can use. So we developed the press pulse therapeutic strategy. And this is the outline for that. And George Yu is an oncologist. Joe Maroon is the team surgeon for the Pittsburgh Steelers, a neurosurgeon, and Dominic D'Agostino, another colleague of mine from the University of South Florida. We put this paper together, and this is an outline of press pulse. The diet is a press. We stress the tumor cells so they can't grow very fast. So we have ketogenic diets and ketone supplementation, maybe MCT oils or some ketones. We keep the pressure on the cells. The patient comes in, he's sick. He's got metastatic cancer, brain cancer, colon cancer. They're all the same. All cancers are exactly the same when it comes to their metabolism. So whatever cancer they have, we put them on the metabolic therapy. They're all stressed out. Cancer creates stress. When you have cancer, you have anxiety. You have impending that you're doomed, maybe. I have something that, especially when it starts to spread, you get all freaked out. So what we do is we use stress management. We have exercise, we have yoga therapy, massage therapy, music therapy, all kinds of therapies that reduce the patient's stress, because when the patient is stressed, cortisol is up, glucose is up. Not good. You got to bring the glucose down, get rid of the stress, put them on these metabolic therapies, and then we pulse with drugs that further target glucose, like insulin and 2-deoxyglucose, and then we use glutamine inhibitors like glutathione, chloroquine and EGCG. I'll talk about that. And then we put them in hyperbaric oxygen chambers. Instead of radiation, we put them in hyperbaric. Hyperbaric oxygen will kill tumor cells as long as you can get rid of the glucose and glutamine. Why you radiate somebody when you can do this? Right? So the patient then starts to feel better. Now, a lot of these people who come in, they're all screwed up anyway. They got diabetes, they got all kinds of metabolic imbalances. All that seems to go away along with their cancer. So then they start feeling better, they get fewer, and then we get resolution. I don't know if we cure the disease. I don't want to say we cure people. I don't know if we cure people. All I know is they're a hell of a lot healthier than they were when they started. How long they live, I don't know. Because we just started this stuff. How many long-term survivors? You know, all I know is many of the ones we treat are living. What am I supposed to say? And some die, okay, because it requires compliance, and sometimes these people are not compliant. And that's another thing. I can't do anything about that. So here's our most recent paper that we just published a couple of months ago out of Egypt, because we can't do this stuff in the United States, you know. We can't do it in England. We can't do this. So this El Saka came to me and he says, I'm using your metabolic therapy to treat glioblastoma patients. I said, wow. So these guys, it's hard to write. I do that for a living. I've written, dozens and hundreds of scientific papers for journals. So, and I know how to organize data, I know how to write the data, and I know how to present the information. The clinicians get the data, we can formulate it, put it in the right order, and then we can publish it. So this is a guy, 38-year-old gentleman. He was a farmer, corn farmer in Egypt. He came in with his whole left side dragging. He couldn't move his arm, he was in bad shape. He was also out of balance in vitamin D. He had elevated triglycerides. He was metabolically a mess, all right? So we put him on the I'll show you what we did, if I have it on the next slide. Well, we took him water-only therapeutic fasting for 3 days. Then he went into a 900 kilocalorie a day for 3 weeks. No, we didn't do any debulking. Then after 3 weeks of this, they took an awake craniotomy and we took the tumor out of this guy. And as soon as we took the tumor out, we brought him back on the metabolic therapy, and we used chloroquine, EGCG, hyperbaric oxygen. After 3 months, he went in for the radiation. And I said, why are you putting him in the radiation? Well, we have to do that because everybody has to do that. You're going to lose your license if you do that. Don't do that. So I said, anyway, we put him on there. He said that he was prepared now. His body was prepared. So look we brought his glucose ketone index down, really good during these treatments. Now here's his tumor in the brain by MRI analysis. You see that white spot? That's his tumor. That was preoperative, before there was any surgery taken, so you can see. And you can see the red line here. This red line is shifted massively. That's what I said. The tumor grows in the brain and causes midline shift. And that's why he's paralyzed. He's getting this part of his brain crushed. So as we began the metabolic therapy, and we did all this metabolic, you can see the shift coming out. It's a combination between the debulking and the metabolic therapy and all the other treatments. So the guy, after 24 months, and he's still going, he went back working in the cornfield. He was doing really well. No steroids. We gave the guy no steroids. So it goes to show you that we can achieve a far better, that's only one patient. Listen, it's not, we got to write more and more of these patients. It just takes time. And then we did it in Turkey for breast cancer. And this was, we have a big clinic in Turkey now that are doing this. And this is a woman who came in. She was also about 30 something, I can't remember. Triple negative breast cancer, which is not good. And it spread to her liver and part of her muscles. And what happens is that we now have management. Look at this. She's doing really well, okay? So what we have the choice. Here's Brittany, standard of care. Pablo, ketogenic metabolic therapy. So how many more Pablos can we get? Cancer is a type of mitochondrial metabolic disease. It's not a genetic disease. This is the singular greatest misunderstanding, and it's the singular greatest tragedy in the history of medicine. This misunderstanding about what cancer is. It's a metabolic disease. It lives on fermentation fuels. If you restrict the two fuels, you can manage any kind of a cancer because they all depend on either glucose, glutamine, or both. And the press pulse metabolic therapy is non-toxic, cost-effective, and in my mind, it'll eventually make all other cancer therapies obsolete. All right? It's just a matter of time because people want to live. They don't want to be poisoned and irradiated. Now, here's all my colleagues from all the different countries that are working with us now. China just came back. They want to dovetail this with the Chinese traditional medicine. And of course, where do you get money to do this? I got to thank Travis Christofferson, author of Tripping Over the Truth. He has a private foundation like your foundation. He collects funds to support my research. Joe Mercola, George Yu Foundation. CrossFit, Greg Glassman of CrossFit is a big fan of what we do. He's been supportive. Dr. Joe Maroon from the University of Pittsburgh. These are individuals giving us money from their pocket. Ellen Davis from her book, My Own University. And I always say, in the past, the NIH, when we weren't really challenging the system, they were very happy to support us. Thank you for your attention.